Hello. Okay. So before we start today, I'm going to hand you out a diagram that we're going to label and work on today, because we're going to be looking at the microscopic anatomy of muscle tissue and how muscles are able to contract in response to motor neuron stimulation. Do you need another one? Yeah. Okay, so we have talked about the different types of muscle tissues previously when we were talking about them when we were talking about different types of tissues. So remember that there's skeletal muscle that is responsible for movement. There's smooth muscle that is responsible for like contraction of blood vessels, constriction of blood vessels, and also constriction and contraction of the bladder. And then there's also cardiac muscle, which is unique to heart. And so we are going to, in this chapter, which is chapter 9 in your book, this primarily deals with skeletal muscle tissue, and this is what we're going to be looking at in lab as well this week. So we can talk about the characteristics of muscle tissue. And so one characteristic of muscle tissue is that it has to be excitable. And this is also a characteristic of nervous tissue. So one neuron can release neurotransmitters that excite another neuron to generate an action potential. And so when we look at this ability to be excitable, remember that we talked about how motor neurons innervate skeletal muscle. So innervate means to make a synapse with. That's two N's, sorry, innervate. Skeletal muscle. And does anybody remember what type of neurotransmitter is released into the synapse? What is the neurotransmitter? It's the A one. Acetylcholine. Okay. So ACH is released. Right? And it binds to receptors. So it binds onto the receptors on the skeletal muscle cells, and then those receptors open ion channels, and this causes the muscle cells to become depolarized. So we'll just put muscles or muscle cells become depolarized. So this is the same idea of depolarization in neurons. It occurs in muscle cells as well. The second thing that muscle tissue has to be able to do is it has to be contractible. So that means that it has to change shape. And one of the things about muscle tissue is that it pulls, but it never pushes. So when we're looking at movement, like raising your arm, for example, that has to do with muscles pulling on the bone, not muscles pushing on the bone. And so this ability to contract will pull on bones and can lead to movement, right? Sometimes when we talk about smooth muscle contraction, remember the muscle kind of contracts on itself. And so contraction can cause vasoconstriction of blood vessels, right? And contraction of the heart can propel blood up and out of the ventricles, through the ventricles to the body, okay? But this idea that it has to be contractible. So we'll talk about why it is contractible in a little bit, okay? The other thing is that it has to be extensible. So this means that it must be able to lengthen. Or stretch. Oops, I am not doing very good with my spelling today. So stretch, the T. Okay. So oftentimes when we do a movement, like say for example, if I lift my leg here, the muscles on my back of my body and my hip have to be able to stretch in order for these to allow to contract. So we have to have ability of the muscle to stretch. So we'll look at that, why that is from a microscopic viewpoint, the individual muscle cells in a minute. And then the fourth thing that it has to be able to do is it has to be able to come back to position. So it has to have elasticity. Oops, elasticity. Okay, so we talked about elastic fibers. So in connective tissue, remember, like there's elastic cartilage in your ear. So when you bend it, it will kind of bounce back. So we don't have elastic fibers in the muscle, but we do have muscles that are kind of spring shaped. And this allows the muscle to come back into its relative length after it has been stretched. So we can say that elasticity is the ability of the muscle to recoil and resume its resting length. So ability to recoil and resume resting length. Okay. So those are the four characteristics of muscle that the muscle tissue has to have in order to function properly. We can also talk about the function of the muscle tissue, and specifically we're going to talk about skeletal muscle here. So skeletal muscle is just one of the three types of muscle tissue. So one function obviously is locomotion. So what would be another example of a function? So movement, locomotion. Boy, and that's not going to work. Sorry, let me erase all this. Okay. So what do you think would be another function? How about thermoregulation? How does it work in thermoregulation? Skeletal muscle. Somebody remember? So when you get really, really cold, what happens? Or if you have a high fever, what tends to happen? You get chills, but what does your body do? Muscle contraction, right? So in thermoregulation, it functions in shivering. So shivering produces heat. Okay. How does it function in joints? So remember that we talked about joints and the role of muscles in the joint. Anybody remember? So this would have been last quarter when we were talking about bones and joints, where bones articulate with one another. So muscles actually help to stabilize joints, right? So we talked about, and we'll talk about more, the rotator cuff muscles that are around the shoulder joint. And sometimes if one of those becomes unstable, if it becomes damaged or it becomes overstretched, or even you can have those muscles that actually pop off of the bone, then your shoulder joint would be less stable, right? So it'd tend to disarticulate, right? So become disconnected. Okay. And then the fourth function is in maintaining posture. So we can say, for example, that there's muscles that allow me to stand upright against gravity, and they have to contract in order to keep myself from falling over, but they're not really actually causing movement, but they're actually just kind of allowing me to stay upright. And so that would be a fourth function of the skeletal muscles. Okay, so this is the first handout that I gave you. So does anybody recognize what bone that is? This is the femur bone. Right, so this could be one of those leg muscles, the quadricep muscles. So this muscle actually has a connective tissue connecting the muscle to the bone. And so what kind of connective tissue is that? What is this connective tissue called, where you have, what connects muscle to bone? Close, not ligaments, but so what's the other one? Tendon, right? So remember that tendons are dense connective tissue, right? So that would be actually just one tendon. That's a dense connective tissue, and they're white because of what? What gives them the white color? It's a protein. No. What gives it its strength and makes it white? Collagen, right? So we have collagen fibers. So that is a structural protein that is found in bone, but it's also found in tendons and ligaments, and that's why it looks white, it's because it has collagen fibers in it. Now, this whole thing is an organ. So one muscle is actually an organ. And so you'll remember that an organ is anything that is composed of more than one type of tissue. So we'll say that it's composed of many different types of tissue. So like your biceps and your quadriceps and your hamstrings, those are actually all considered to be separate organs. And one thing that's not shown here is that skeletal muscle has nerves that are supplied to it, and it also has blood vessels. So we have nerves coming into the skeletal muscle. We have blood vessels. And the blood vessels are composed of smooth muscle. They're composed of connective tissue. They actually have their own little nervous supply, right? So that would be part of the muscle itself, would be the nerves and the blood vessels that are serving it, because the blood vessels are bringing in oxygen for ATP production and then getting rid of CO2, which is a major waste product of cellular respiration. Okay, so sometimes you hear people talking about muscle fascia. And so the muscle fascia is believed to be really important because it allows the muscle to move within a connective tissue sheath. And so when you get a massage, for example, the connective tissue, when you massage it, actually kind of stays loose. And so it actually prevents adhesions from forming in the muscle itself, and it keeps the muscle kind of healthy, right? So massaging muscles is a good thing. So when we look at this fascia, this is called the epimysium. So that MYS means muscle. That is one of the root words that means muscle. And I always remember epi means upon. And so this is the outer connective tissue sheath, okay? You can also see, at least in this diagram, not on your diagram, that there's actually veins and arteries that are coming into the muscle. So that would be the blue and the red on this diagram. Okay. So the muscle itself consists of fascicles. So this would be an example of one fascicle. Okay, so that's one bundle of skeletal muscle fibers. Okay, so the skeletal muscle fibers are bundled together into fascicles. And the connective tissue sheath of this fascicle is what is referred to as the perimysium. So think peri means around. So that is around the different fascicles. So sometimes when you tease apart meat, you know, it forms these strands, right? And those are probably the fascicles. If you think about like if you're teasing apart pork, sometimes they come apart in these, you know, little pieces, and those would be the fascicles. Okay, so this right here is one muscle fiber. And this is actually one muscle cell. So it's kind of confusing because you can see when you look inside that muscle cell that it looks like there might be more than one muscle cell, but this is actually the cell. And the muscle cells are very interesting, the skeletal muscle cells, because they are multinucleated. And so this is because during the development of skeletal muscle, the stem cells fuse. So many stem cells fuse together to form one muscle cell. So we'll say that that muscle cell is multinucleated. So this is multinucleated. That's with a C, okay? So the nuclei, you'll notice here, are kind of pushed to the edges of the cell. So this would be a nuclei, this would be a nuclei, right? So those are the nuclei. So what this suggests to me is that there's something in the cell that is really important, right? That is taking up the majority of the space, and everything else is just kind of pushed to the edges. And what is really important in muscle cells are what are called myofibrils. So one little thing of that is called a myofibril. So like MYS, whenever you see myo, the myofibril, that myo means muscle. So the myofibril is an organelle inside the cell. So remember that organelles are bound by a plasma membrane. And inside this organelle, we have myofilaments. So it contains myofilaments. And myofilaments are molecules and they're proteins. And so the myofilaments, does anybody know what they're called? What are the two myofilaments that you have inside your muscle? Anybody remember? Carly should know this. Majors biology. Actin. Have you ever heard that word before? Yeah. Okay. And myosin. Okay. So these are protein. So if you wonder why muscle is high in protein, this is the reason why. So like if you're like craving protein, right, you eat muscle, right? You can also eat vegetables, there's protein in nuts and stuff too. But protein, but muscle tissue is high in protein because of these actin and myosin filaments, which you'll notice fill up most of the cell, right? Because they're contained within the myofibrils. Okay. So that was part of the microscopic anatomy, but there's a little bit more that I want to tell you about. And so again, one muscle fiber is one cell. So remember we talked about fibers in terms of neurons. And so what was the fiber of a neuron? What was that actually? So we talked about nerve fibers. What were they? So if I have a nerve and it's filled with fibers in it, what is that? Close. So that's where the nerves actually branch apart and come back together. But a nerve fiber is actually the myelinated axon, right? So the axon, a nerve fiber is actually the axon that comes out and goes to and innervates the different organs, okay? So when we look at a muscle cell, skeletal muscle cell has what is called a sarcolemma. So another word, if you think about sarcophagus, that's flesh eating. So you can have sarcophagus beetles that eat flesh, right? Sarco means flesh, and lemma means husk. And so this is the outer covering, covering, or plasma membrane of the cell. And it is also called the endomysium. So it has two names, sarcolemma and endomysium. So in that previous diagram, if you get that diagram back out, the plasma membrane of a muscle fiber is called a sarcolemma or the endomysium. So we have peri, we have epi, and we have endo. So if we go back to that previous diagram, okay, this would be the endomysium. Okay. Also called the sarcolemma. So the other important parts, besides we already mentioned the myofibrils, right? So the sarcolemma, the myofibrils, which are the organelles with filaments. The other important structure is what is called the sarcoplasmic reticulum. So the sarcoplasmic reticulum is a specialized organelle. You've heard of reticulum before when we talked about cell biology. That would be like the endoplasmic reticulum, the smooth and the rough endoplasmic reticulum, right? So this is an organelle, membrane-bound structure inside the cell that contains or stores calcium ions. So we're going to learn that calcium ions are really important for muscle contraction. So if your calcium is too high or too low, it can lead to problems with your muscles, just like with your nerves. The calcium is used, remember when we talk about the release of the neurotransmitter by the neuron is induced by the influx of calcium ions. Okay, so look at your diagram again, or on the second page, okay? So one of the things about skeletal muscles is that they are striated. So we can look at the striations. Now the striations have to do with the arrangement of actin and myosin. So that's arrangement, sorry, M-E-N-T, of actin and myosin. So this is, these muscle cells can be really long, and so they're not showing the whole muscle cell here because it can be really long, right? In the big muscles, they can be very long. So this is a muscle fiber. And notice the banding pattern. So the dark band is called a A band. So they use A, the letter A, that's the dark band. It has both actin and myosin. So it is darker in coloration, actin and myosin. And then we have the I band, and I'm not sure why they gave them these letters, but they're not very interesting. We have the I band, which is light in color, and it contains only myosin. So there's something about the arrangement of the actin and the myosin that gives it this banding pattern, which is what is referred to as the striations. So what are these purple things? What are they? It's pushed to the outside. What's pushed to the outside? No. That's the nucleus, right? Notice how there's more than one nucleus. So these would be the nuclei. Okay. So if we go down to the next picture, right? This is the outer plasma membrane, which is called the sarcolemma. Right? This right here is a myofibril. Now notice they also are banded, right? They have, if you took a myofibril out, you would see the banding. So remember the actin and the myosin are in the myofibrils. And then if you look at the next image, it shows the sarcoplasmic reticulum. So I screwed up and I didn't use the unlabeled one. So this is, do you guys have this image? Let's see. No, you don't. Okay. We'll skip that for it. Come back. Okay. Okay. So you have this image, right? Okay. But you don't need to know all this, so don't start writing it down, okay? So when we looked at the actin and myosin, the actin is the thin filament. So right, actin, thin filament. Right? So where you see the thin filament right here, that would be actin. Okay. So in this diagram, it's blue, but in yours, it's just a black. But notice how it's thin. The myosin is the thick filament. Okay, so in the red here, but the thicker filament would be myosin. Okay. So notice right here where there is only actin, so there's no overlap between the bands, that would be lightly colored. And so that is the I band. So you can draw that one on there. The I band is the lightly colored one. Okay. And where you see overlap between the actin and the myosin, that is the A band. So that would be in the middle here. That's the A band. So there's overlap. So if you go down to the next image, it should look something like this, right? So again, this would be actin. The red here, the thicker ones would be the myosin. And then there is another type of filament besides actin and myosin, which is important, and this is called elastic. So this is the elastic. The one that looks like the spring is elastic. So you can imagine that because it has this springy shape, that if I stretch my muscle beyond its point of like being happy, it's going to spring back, right? So the muscle has elasticity in it. And then the other thing that you need to know in this diagram, these are Z discs. And so this is what the muscles, this is what the muscles pull against, right? So the muscle itself has to have something to pull against. And so these right here are structure. It's a structure, it's like scaffolding in the muscle. And that is what is a Z disc is called. Okay. Now, this one unit from one Z disc to another is called a sarcomere. So a sarcomere, right, is in the myofibrils, right? And it's the arrangement of actin and myosin, but it is from one Z disc to another. And it is the unit of shortening or contraction. Shortening via contraction. Okay. Okay, so how many of you, this is like the very first time you've ever heard any of this information? Okay, so half of you. Okay, so when I was in school, like I had to go through like 3 or 4 different classes of the teachers presenting this information before it finally started to sink in, right? So generally, if you're a biology major, right, you have like 3 or 4 classes. Like you'd have zoology, you'd have general biology, then you'd have zoology, and then you'd have maybe anatomy and physiology. And so every time they just kind of keep repeating some of the same information until it starts to stick in your brain. Yeah.
Someone asked me this the other day, what can you do with just plain biology?
It is almost impossible to just get a job if you're going to do research, you might be able to get a job in the lab. If you're going to be like, you could be a field biologist, but generally that is just temporary seasonal work where you would help people with their research projects.
So really like
Yeah, so generally you could get a job in a lab, like if you wanted to, but it's not generally what's required. So if you wanted to work at Interpath Lab or whatever, yeah. So generally you have to go beyond that. You might be able to get a job with a non-profit organization, right? Yep.
Is this like micro too? Like so when I take micro next term?
No, no, they don't talk about it in micro. Yeah, okay. So there is another image, I believe, which is a cross section, and I don't have it here on mine, but that first one, so that first circle, that would be actin only. So notice how that is showing a line. So if I looked at a cross section, that would be actin, right? You don't need to know those second two. Actually, the third one is actually the Z disc. So you could write Z disc on the third one. And then the last one is myosin.
The last one is myosin?
Yep, the last one is myosin. So notice, wait a minute. Am I wrong? I think they have it pointed to the wrong thing. Oh, the last one is myosin and actin, sorry. Okay, so sorry, I start over. The first one is actin only. The second one is myosin only. The third one would be the Z disc. And then the fourth one is actin and myosin. So they're showing a cross section. Sorry about that.
Is it with a disc with a K or a C?
It is with a C. Okay. Okay, so how does this all come together? So the people that figured this out was like in the 1960s, right? They came up with a model. And so in biology, we have models for all kinds of things. But one of them is the model of contraction. And before this model, what they thought happened was that the proteins themselves contracted. So like maybe the filaments were able to shorten, the individual filaments were able to shorten and lengthen, but you couldn't really see how a protein would be able to do that very well. So this is the new model for it, and they won, I believe, the Nobel Prize for it, the scientists who put it forth. And this is the idea that the contraction actually has to do with not the shortening of the filaments, but the way that they slide together. And so you can think of it as kind of coming together, and the actin and the myosin coming and shortening, and the sarcomere, and then sliding back apart, right? So in order to do this, when we look at the sliding filament model, we have to look at how the actin and the myosin would interact, okay? So on the actin, we have what are called myosin binding sites. So the myosin can bind to the actin. On the myosin, we have myosin heads. When the myosin heads bind to the myosin binding sites, that is called a cross bridge. So if you combine these, right, you get what is called a cross bridge. So the myosin heads bind to the myosin binding sites, okay? The myosin heads are also able to pivot. So I'll put down here, able to pivot and cause the sliding together of the filaments. This allows the sarcomere to shorten and the muscle to contract. Okay. So the myofilaments themselves stay the same length. It's just the way that they slide against one another that actually causes the muscle to shorten. So if we look at a diagram or an image from your book, I didn't give you this one. So this is where the muscle is not shortened and the sarcomere is pretty wide, right? So these are the myosin heads, these little things that stick up, right? So those are the myosin heads, and they're going to bind to the actin, and then those myosin heads are going to pivot, and then notice how they cause the sarcomere to shorten as the filaments slide closer together. So the distance between the Z discs becomes smaller and the sarcomere shortens. So this would be relaxed, and this is when the muscle contracts. Okay. So we need to ask why or how do we regulate when muscles contract and when they don't, right? So how can we cause a muscle to contract versus allowing it to relax? And so we're going to talk about the events leading to contraction. So the most important thing to realize here, I think, is that there are regulatory proteins associated with the actin filament. The first one is a protein called tropomyosin. So this is another protein separate from the actin and the myosin called tropomyosin. And tropomyosin blocks myosin binding sites. Right? So this would prevent contraction. Okay, so that's the first regulatory protein. The second regulatory protein is called troponin. And this is smaller than the tropomyosin, and it actually doesn't bind to actin, but it binds to tropomyosin. So this binds to tropomyosin. Okay. It also binds calcium. So calcium ions have a plus, calcium ions have a +2, right? So they have +2 charge, they're an ion. Okay, so the first thing that has to happen for contraction to occur is that we must have ACH binding to receptors on the sarcolemma. And remember that acetylcholine acts directly. So these receptors are also ion channels. So what kind of channels do you think that they open up They open up the same ion channels as they do in neurons. So what is it? Is it sodium, potassium, chloride, calcium? What kind of channel do you think they open? What channel causes depolarization? Sodium, right? So sodium channels open. Depolarization occurs. Okay. So this is the same type of depolarization. It's a change in the membrane potential, and the membrane changes and becomes slightly positive charged on the inside, right? And this depolarization is really important because it is what leads to the release of calcium ions. Okay, so the second thing that happens is that the depolarization causes the sarcoplasmic reticulum, this is abbreviated as SR, the sarcoplasmic reticulum, the SR, to release calcium into the cytoplasm of the cell. So it's released not to the outside, but into the inside. So just to release calcium ion inside. There. Okay. Okay. Third thing, calcium binds to troponin. That calcium binding to troponin causes the protein, troponin, to change shape. And remember that troponin is bound to tropomyosin, so it eventually causes tropomyosin to change shape as well, right? So what happens here is that binds to troponin and then causes tropomyosin to change shape. When it changes shape, it moves. I'll put move. Okay, so it changes shape, it moves. And then it exposes the myosin binding sites. Okay. So what this means is that contraction can only occur if calcium is released. Yes.
Is there a difference between tropomyosin and
Tryptomycin or tropomyosin? What tryptomycin?
Yeah. Is there such thing called tryptomyosin?
There's tryptophan,
Maybe that's
which is an amino acid. Yeah. I don't know. I'd have to look up tryptomycin. I'm not sure what tryptomycin is. That does sound familiar to me, but that's not related to this.
awesome.
Okay, so let's look at how this works. So I didn't give you the handout, but there's a handout in your book, right? So this would be the motor neuron. That is part of the somatic division of the peripheral nervous system, right? It releases ACH into the synapse. Depolarization occurs, and that depolarization goes down into the inside of the cell. You don't need to know about those T channels, and it causes calcium to be released. Okay, so that would be the first part. And then in the second part, what happens? Skipped ahead here. Okay, so in the second part, what happens is we have these proteins that are regulatory. So notice the tropomyosin is long strands, right? So notice that right here, the tropomyosin is blocking these little holes, and those holes represent myosin binding sites. So this would be without calcium present. The yellow is the troponin. Right? So if calcium were to bind to that troponin, tropomyosin is going to move, and then those binding sites are going to become available. So in the next image, we see this. Okay, so calcium ions are released. So these are the calcium ions. Notice that they bind to troponin right here, right? Tropomyosin moves, and then we get a cross bridge formation. So notice how the myosin then is able to bind to the actin. And notice that it has this long arm, the myosin head does. And when the myosin head binds, it then subsequently pivots, and it causes the actin filaments to slide in relationship to the myosin filaments, and it causes a shortening of the sarcomere. Okay, so it's pretty amazing that somehow these scientists were able to figure out all of this mechanism, right? This obviously is not something that you're actually able to visualize. It's something that you actually have to, we might be able to see it with an electron microscope, some of these, but it's not something that we can see with our naked eye, these things happening. Okay, so when we look at energy, okay, so ATP is the energy source for muscle contraction. And we'll talk about the different muscle fibers like slow twitch, fast twitch, and oxidative versus glycolytic fibers that are involved in exercise. But right now, we'll just talk about the role that this energy plays. Okay, so ATP binds to the myosin head, or the myosin heads. It is broken down. So ATP is converted to ADP, that's adenosine diphosphate, right? Plus P plus energy, right? So what this energy does is this energizes the myosin head, right? So this energizes the myosin head. The myosin head then binds to the actin. Binds to actin. And it releases that energy by pivoting, right? It pivots and uses stored energy. So what we've essentially done is that we've taken chemical energy. So this would be like right here. This is chemical energy. Right? And we've actually turned this into, when it pivots, that would be mechanical energy. Because it's doing work, right? And it's doing physical work. So we know from thermodynamics that when you convert from one form of energy into another, it's inefficient, right? And so this pivoting, this conversion of chemical energy into mechanical energy, it produces as a waste product what type of energy? When you exercise, what energy do you release into the atmosphere? Nope. So what happens when you're working out? And you get hot, right? Okay, so this is heat energy, right? Heat energy is lost. So this is why when you exercise, you get hot, right? Because you're converting from chemical to mechanical. So when you convert from one form of energy to another, some of that energy is always lost as heat, right? What heat is just molecular movement, right? So when you heat up, the molecules in your body are just moving faster, right? Too much of that energy and your proteins start to denature, right? You get too hot. So you heat it up and then you cause it to be released into the atmosphere via evaporative cooling, right? Or radiation, you radiate heat. Okay. Okay, so the other thing that's important to realize that for some reason, and I'm not sure why this is the case, let me go back. In order for the myosin head to unattach from the actin, another ATP molecule must be bound, okay? So I'll put 4 myosin to unattach from actin. Another ATP must be bound. Okay. So when people die and they start using up their ATP before decomposition sets in, so before you start to rot after death, one of the things that happens is rigor mortis. Right? So this is where you get stiff upon dying, right? And then that stiffness can last for hours, right? But eventually it will go away because the cells will start to decompose, right? But this rigor mortis is due to the fact that you run out of ATP and the myosin heads stay attached. And so the muscle stays contracted, right? So run out of ATP and the muscles stay contracted.
How long for the rigor mortis? How long
I don't know, because I think it probably depends upon like how warm, like if it's outside or if it's, in cold temperature, it would last a lot longer, right? So it just depends upon when decomposition will start to
Because I know we had a horse die one time, and like as soon as she hit the ground, like 2 or 3 minutes later, rigor mortis had set in.
Right.
And the leg just like stick it straight out.
Right.
And so I thought.
I wonder how long. Do you know how long it lasted? Did you?
I don't really know, but I
Okay.
just know
I think it's just hours. Yeah. So we don't have a lot of ATP stored up.
Mhm.
We are constantly producing it, and so almost immediately people will start to get stiff because of rigor mortis. Okay. So in your book, there's a little diagram that shows, again, ATP in this particular instance. So notice how ATP binds here. If we just start here, I'm actually going to start at number 4. But the energy, the head gets a little energized and it kind of pivots. Then when it binds, right, it pivots, releases that energy. And then in order for it to release the ATP, release its bound, the myosin and actin, another ATP must be present. And so this just shows the role of ATP in this process of muscle contraction. Okay, so I'm going to show you a short little video that animates this and talks kind of as a review of all the stuff that we've just talked about.
Muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T-tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
Okay, are there any questions about that? It was a pretty good review. Okay. No questions?
How quickly does that happen?
Milliseconds.
Hello there.
Oops.
You caught me while I was working.
The Khan has a crash course in biology. He's pretty funny. Khan Academy. Okay, so that is all I'm going to talk about today. Yes, I figure your brain is like, what up?
When there's only like 4 of us here, it feels like I don't know why.
It's harder.
It feels longer. Like the class period